Okay, let us start our today's topic then. So we were discussing about prokaryotic cell. We discussed the cell envelope. We discussed the genetic material. We discussed ribosomes. We discussed the inclusion bodies, right? Then we are now to discuss about the structures which are present extracellularly but originate intracellularly. As I told you in the last lecture, that a prokaryotic cell may have a flagellum or may not have a flagellum. If a prokaryotic cell is without a flagellum, we call that cell as atrichous. If a prokaryotic cell is without a flagellum, we call it as atrichous. If a prokaryotic cell has got single flagellum, we call it as monotrichous. If prokaryotic cell has got two flagella at two opposite poles, we call it as amphitrichous. If a prokaryotic cell has got a group of flagella at one end, we call it cephalotrichous. If a prokaryotic cell has got two groups of flagella at two opposite poles, we call it as lopotrichous. And if a prokaryotic cell is all around surrounded by flagella, then we call it as peritrichous. Now today we are going to look at the structure of flagellum of a prokaryotic cell. The flagellum of a prokaryotic cell is formed of three parts. It is formed of a filament. It consists of a hook, and it also consists of a basal body. Basal body. The flagellum of prokaryotic cell is formed of three parts. A long straight part known as filament. This filament, it is the long straight part. This filament, it may be approximately 1 to 70 nanometers in length. This filament, it is formed of a protein known as flagellin protein. It is formed of flagellin protein molecule. So this is the longest part of the flagellum. Filament is the longest part of the flagellum. This filament, it may measure 1 to 70 nanometers in length. It is formed mainly of a protein known as flagellin protein molecule. These flagellin protein molecules, they are helically arranged and they form a tubular hollow filament, right? So they form a tubular hollow filament. Then attached to this filament is a short and curved hook. This is the short and curved hook. Flagellum is attached to a short and curved hook. This is hook. This hook measures some 45 nanometers in length. Then hook is attached to a basal body. Hook is attached to a basal body. This basal body, it is formed of 2 or 4 rings. It is formed of 2 or 4 rings. In gram positive bacteria, there are 2 rings. In gram positive bacteria, there are 2 rings. In gram negative bacteria, there are 4 rings in basal body. There are 4 rings in basal body. So this is the basal body of a gram negative bacteria. It has got 4 rings. One ring is known as L ring, another is known as P ring, another is known as S ring, and another is known as M ring. So this basal body of gram negative bacteria is formed of 4 rings. These rings are known as L, P, S, and M. This L ring, it is embedded in lipopolysaccharides. L ring is embedded in lipopolysaccharides. P ring, it is embedded in peptidoglycans. L ring is attached to lipopolysaccharide count, right? P ring is attached to peptidoglycans. SM and ring, sorry, SM M ring, S and M ring, they are present in cell membrane. They are present in cell membrane. So the basal body of a prokaryotic cell, it may have 4 or 2 rings. In case of gram-positive bacteria, there are only 2 rings. In case of gram-negative bacteria, there are 4 rings. The 4 rings of a gram-negative bacteria, the 4 rings of a gram-negative bacteria, they are L, P, S, and M. L is the outermost ring. L is the outermost ring. It is present in lipopolysaccharide covering. It is present in glycocalyx, right? P is the peptide. P ring is present in peptidoglycans. It is present in. It is embedded in cell wall. And SM and M rings, they are embedded in the cell membrane, right? So these are the 4 rings of gram-negative bacteria. What about the 2 rings of gram-positive bacteria? These 2 rings of gram-positive bacteria, they are SMN. Sorry, they are SMN, S and M. They are both attached. Sorry, S is present in peptidoglycans and M is attached in cell membrane. Right? So in gram-negative bacteria, there are 4 rings. In gram-positive bacteria, there are 2 rings. The 2 rings of gram-positive bacteria, outer ring is embedded in peptidoglycans and inner ring is embedded in cell membrane. While as these 4 rings, outer ring is attached to lipopolysaccharide membrane, inner to that P ring that is embedded in peptidoglycan, and S and M, they are embedded in cell membrane, right? So this is the structure of flagellum of a prokaryotic cell. This is the structure of flagellum of a prokaryotic cell. So how does this flagellum rotate? This flagellum rotates by the movement of these rings of the basal body. In these rings, there are proton motors. In these rings, there are proton motors. Those proton motors, they make movement by the flow of protons through them. And when protons flow through them, energy is released. By the help of that energy, these rings rotate. When the rings rotate, they make this hook to rotate. And hook makes filament to rotate. So in this way, this flagellum makes movement by the help of this basal body. The rings contained in basal body, they are having proton motors in them. And proton flow makes the energy available for the movement of the flagellum, right? So this is the structure of the flagellum of a prokaryotic cell. Clear? After this, we will look at some other structures. Now, a prokaryotic cell may bear sex pili on it. A prokaryotic cell may bear sex pili on it. These sex pili, they are usually found. They are found in sexually reproducing gram-negative bacteria. Gram-negative bacteria. Negative bacteria with fertility factor. With fertility factor. So these sex pili, they develop in sexually reproducing gram-negative bacteria with fertility factor. So those gram-negative bacteria which have got fertility factor in them, they develop these sex pili. So what are these sex pili? They are hollow tube-like outgrowths of the cell envelope. These are hollow tube-like outgrowths of the cell envelope. They are short structures. They measure just 8 to 20 microns in length. They are very short tubular outgrowths. They measure 8 to 20 microns in length. And they are formed of a protein known as pillin protein. They are formed of a protein known as pillin protein. Flagella was formed of flagellin protein. These pili, they are formed of a protein known as pili protein. How many sex pili develop on the bacterial cell? On a bacterial cell, there may be just 1 to 4 sex pili. On a bacterial cell, there may be just 1 to 4 sex pili. So their number is limited. Their number is limited. The number of sex pili on a bacterial cell may be just 1 to 4 per cell, right? What is their function? They form conjugation bridges. They form conjugation tubes. They form conjugation tube or conjugation bridge between two sexually reproducing bacteria. Between two sexually reproducing bacteria. What is exchanged through these sex pili? What is exchanged through these sex pili? Through these sex pili, the bacteria exchange the plasmids. Bacteria exchange the plasmids, right? So when it joins with another bacteria, it forms the conjugation tube. Then through this conjugation tube, plasmids are exchanged between the bacteria. And this exchange of plasmids in bacteria, exchange of gene segments between two bacteria, it helps them in their survival. It induces variations in bacteria. And by the help of those variations, the bacteria may survive in changed environmental conditions. So conjugation is very much essential for bacterial survival. Sexual reproduction is very much essential for bacterial survival. Because through this sexual reproduction, they exchange gene segments that induces variation in them. And through that variation, they can adapt to changed environmental conditions. So that is the function of sex pili. So they are only limit a few number 1 to 4 per cell. And they are hollow tube-like outgrowths, measuring just 8 to 20 microns in length. And they help in the exchange of gene segments. They are not found in all bacteria. They are only found in sexually reproducing gram-negative bacteria with fertility factor. Similarly, then we have got one more structure which is present extracellular. That is known as fimbriae. Okay. Now fimbriae. These fimbriae, they are again hollow bristle-like structures. Bristle-like structures. They are hollow bristle-like structures, which are present on the cell envelope of some prokaryotes. These fimbriae, they are 300 to 400 per cell. The number of fimbriae is some 300 to 400 per cell. And they are shorter than sex pili. They measure just 0.15 to 1.5 microns. They are much shorter than sex pili. They are much shorter than sex pili. They measure 0.15 to 1.5 microns in length. So what is their function? What is the function of these fimbriae? These fimbriae, first I will tell you, they are formed of a protein. They are formed of a protein known as curlin protein. Fimbriae are formed of a protein known as curlin protein. And these fimbriae, they help the bacteria in attaching to the substratum and in adhering to each other to form colonies. So they help them in attaching to substratum. They help bacteria attaching to substratum and to each other. And to each other to form colonies, to form bacterial colonies. So they serve as anchorages. They serve as anchorages. They help the bacteria to attach to the substratum to get adhere to the substratum, and they also help the bacteria to get attached to each other, so that bacteria accumulate at a single place in large numbers to form bacterial colonies. So that is the function of fimbriae. So these fimbriae, they are hollow bristle-like structures, 0.15 to 1.5 microns in length. The number of fimbriae per cell is large. It is 300 to 400 per cell. These fimbriae, they are formed of a protein known as curlin protein. And the main function of these fimbriae is to help the bacteria to attach to the substratum and to form colonies. So that is all about the structure of prokaryotic cell, right? After prokaryotic cell, now we have to discuss the eukaryotic cell. Now we have to discuss the eukaryotic cell. Eukaryotic cells. What is an eukaryotic cell? An eukaryotic cell is a cell with true nucleus. Eu means true. Eu means true. And karyon means nucleus. So they have got a true nucleus. What is a true nucleus? A true nucleus is a nucleus which is covered by a double nuclear membrane and having linear chromosomes with linear DNA in them, right? A true nucleus is a nucleus covered by a double nuclear membrane and having linear chromosomes with linear DNA in them. So these cells, they have got a true nucleus. That is why we call them as eukaryotic cells. As I told you, these eukaryotic cells, there are two types: plant cells and animal cells. Why we classified eukaryotic cells into two types? We classified eukaryotic cells into two types because these plant cells, in addition to cell membrane, they have got a cell wall also. Plant cells, in addition to cell membrane, they have also a cell wall covering the cell externally. Whereas this cell wall, it is absent in animal cells. Animal cells only have plasma membrane. In addition to that, these plant cells, they have got structures, organelles known as plastids. Those plastids are absent in animal cells. In animal cells, there are well-developed lysosomes. In plant cells, those lysosomes are not well developed. Some authors consider that plant cells do not have lysosomes. Some authors say that plant cells have very small lysosomes, right? So there are some differences between plant cells and animal cells. So the first difference, just the first prominent difference, is the presence of cell wall in plant cells and its absence in animal cells, right? In plant cells, there are plastids. In animal cells, the plastids are absent. In animal cells, there are lysosomes. In plant cells, the lysosomes are absent, right? Then there are some more differences, right? So we will discuss first the cell wall of a plant cell. We will first discuss the cell wall of a plant cell. So let us discuss the cell wall. This cell wall of plant cell, it is mostly formed of. It is mostly formed of cellulose. It is mostly formed of cellulose. And its thickness varies in between 0.5 to 10 microns. Its thickness varies in between 0.5 to 10 microns. This cell wall, it is laid in two layers or three layers. Mostly it is present in two layers. One layer of the cell wall is known as primary cell wall. Primary cell wall. And another is known as secondary cell wall. So first primary cell wall is formed, then it is followed by secondary cell wall, right? In some cases, in only a few cases, a tertiary cell wall may be also formed. Tertiary cell wall may be also formed. Usually the cell wall of plant cells is of two layers: primary cell wall and secondary cell wall. But sometimes a third layer, tertiary cell wall, may also be formed, right? We have to look into the structure of this cell wall. How this cell wall is formed. We have to see how this cell wall is formed. So, when an animal cell divides, it breaks into two daughter cells. Suppose this is an animal cell. It undergoes cell division. This is animal cell. It has to undergo cell division. It passes through mitosis or meiosis. So this cell elongates. It develops a cleavage furrow in the middle. The cell develops cleavage furrow in the middle. And then finally, the cell breaks into two daughter cells. This parent cell breaks into two daughter cells. Right? These are the two daughter cells. Right? What happens to a plant cell? How a plant cell divides? This is a plant cell. How does it divide? An animal cell, at the time of cell division, when it completes the cell division, the last phase of cell division is cytokinesis, right? The cytoplasm is divided. In the phase of this cytokinesis, this cell elongates. This cell elongates. It elongates in length. And in the process of elongation, it develops a constriction in the middle. It develops a constriction in the middle. Finally, the plasma membrane of the two sides, it meets and it breaks the cell into two daughter cells. So what happens in plant cells? You know plant cells are externally covered by cell wall. This cell wall is very rigid. Cell wall is a hard, rigid structure. So how do plant cells divide there? Do plant cells divide? Do plant cells divide? Plant cells divide. Plant cells show mitosis as well as meiosis in certain cases, right? So how these plant cells divide? So these plant cells, they do not divide by forming a constriction. Instead, what happens? In the center of the plant cell, at the time of cell division, a partition is laid. This partition, it is known as middle lamella. This partition is known as middle lamella. So at the time of cell division, in the center of a parent cell, this is a parent cell. In the center of a parent cell, a partition is laid. As we divide rooms in our houses, if we have a large room and we want to divide it into two, how do we divide that large room into two? We lay a partition in between at its center, right? So similarly, this plant cell, for the sake of division, it lays a partition in its middle, in the middle of cytoplasm. This partition is known as middle lamella. How this partition is laid? This partition is laid by the help of Golgi bodies. This partition is laid by the help of Golgi bodies, right? So Golgi bodies, they synthesize calcium and magnesium peptide. They synthesize calcium and magnesium peptides. Magnesium peptides. These are polysaccharides. These are Golgi bodies. They synthesize calcium and magnesium peptide. And this calcium and magnesium peptide, it is laid in the form of a layer. So this layer of calcium and magnesium peptide, which divides a parent and plant cell internally into two compartments. This is known as middle lamella. So cytoplasm of the plant cell is divided into two compartments by the formation of a middle lamella in between them. Then on the two sides of middle lamella, a primary cell wall is laid. On the two sides of middle lamella, a primary cell wall is laid. So primary cell wall is laid on this side and primary cell wall is laid on the other surface of middle lamella. So this dotted line, it indicates the laying of primary cell wall. So this is primary cell wall. Primary cell wall. Of what material this primary cell wall is formed of? This primary cell wall, it is mainly formed of cellulose microfibrils, which are laid in a gel-like matrix of hemicellulose, and then it is strengthened by some pectin fibers. So first of all, for the laying of primary cell wall, cellulose microfibrils are formed. Cellulose microfibrils are formed. And these cellulose microfibrils, they are laid in the form of a network. For the formation of primary cell wall, the cell first synthesizes cellulose microfibrils. You know that cellulose microfibrils, they are synthesized from beta glucose units. We have discussed the formation of cellulose. So long beta glucose units, beta glucose residues, they form long chains. Then many such long chains combine and they form the cellulose microfibril. Where these cellulose microfibrils are laid, they are laid in a gel-like matrix of hemicellulose. So on the surface of middle lamella, this cell, it synthesizes hemicellulose. It synthesizes hemicellulose. And in this hemicellulose is a gel-like material. In this gel-like material, the cell walls are cellulose microfibrils are. This is hemicellulose. This hemicellulose is a gel-like material. It is gel-like material. This gel-like material, hemicellulose, it is synthesized by the cell itself, cytoplasm of the cell. So first this gel-like matrix is formed. Then in this gel-like matrix, cellulose microfibrils are deposited. Cellulose microfibrils. Cellulose microfibrils are deposited. Then these cellulose microfibrils, they are strengthened by pectins. They are strengthened by pectins. These pectins, they are the polysaccharides again. So these are the pectins. These pectins, they strengthen the cellulose microfibrils, and then short peptide chains, they weave through this cellulose microfibril. They hold the cellulose microfibril. They tie up the cellulose microfibril. Do you understand what I'm saying? So for the formation of primary cell wall, a cell first lays gel-like matrix of hemicellulose. We call this hemicellulose as xyloglucan. Xyloglucan. Why this hemicellulose is also known as xyloglucan? Because it is formed of sugars, xylose and glucose. Hemicellulose is formed of sugars, xylose and glucose. That is why hemicellulose is also known as xyloglucan. So it is a polysaccharide. Hemicellulose is a polysaccharide. But this polysaccharide, it is in a gel, it has got a gel-like consistency. It has got a gel-like consistency. So on the two surfaces of this middle lamella, this hemicellulose is secreted. This hemicellulose is secreted. Then in this hemicellulose, cellulose microfibrils are laid. Then in this hemicellulose, cellulose microfibrils are laid. Cellulose microfibrils, they are laid in the form of a network. They are laid in the form of a network. So these cellulose microfibrils, they are laid in different directions. And these cellulose microfibrils, they form a network. Then this network is strengthened by these cellulose microfibrils. They are tied up together by pectin chains. Cellulose microfibrils are tied up together by pectin chains and by short peptide chains. So cellulose microfibrils are tied up together by pectin chains and short pectin chains. And this whole mixture of hemicellulose, cellulose microfibrils, pectins, and peptide chains, it forms the primary cell wall. So of what material now primary cell wall is formed? So primary cell wall is formed of hemicellulose. Hemicellulose is gel-like material. In this gel-like material are laid cellulose microfibrils in different directions in the form of a network. Then cellulose microfibrils, they are joined together. They are weaved together by pectins and peptide. So then this whole structure, it solidifies and it becomes the primary cell wall. So this is the structure of primary cell wall, right? So our time is running out. This is sufficient for today. Thank you for being with me. Okay, bye.